Hey everybody, I hope you're all having a safe and fun 4th of July. As promised, I still want you to be able to have the lecture experience that you signed up for in taking this class, especially for this unit. This is a really fun unit. I think you guys are going to have a lot of fun learning about the brain and how it works, but I just feel like the reading on its own, even with some helpful videos, can maybe be a little bit complicated or maybe feel like a lot. So I just want to make sure that you are having the most options for your learning experience. So we're going to go through our normal lecture that we would normally do on Saturday. So this is the biopsychology unit. We're going to talk at the beginning about basically the nuts and bolts of how the brain works, specifically how our neuronal systems work. And then after we get through all of that kind of base foundational information about how our brain communicates, we're going to move into more applied stuff. So specific lobes of the brain and what they do and what that looks like in real life. So I think these are both really fun lectures. So by all means, pause, rewind, take notes, go at your own pace. If you need to take a break, then that's fine. And we'll just dive into the wonderful world of biopsychology together. So to get started, like I said, this first part of the lecture, we're going to be focusing on neuronal communication and the nervous system and endocrine system. So these are really the baseline, background, foundational parts of our brain and body and how they communicate together. So how does the brain work? How does it send messages? What types of messages are there? Okay, so we're going to start with neuronal communication, which entails kind of understanding how the brain sends messages to and from different regions of the brain in order to relay that really critical information about your body and the surroundings that it's in. So we start with the specific cells that make up the nervous system. So the two main types of cell types that make up the nervous system are glial cells and neurons. Okay, so neurons are the stars. They're the most important parts. And then the glial cells will form sort of supporting roles to the neurons. So you can see here in this image, we have a neuron, that's the yellow part here. And then these blue and pink things that are around it are different types of glial cells. So the red ones are astrocytes, and here you have oligodendrocytes. You don't need to know that. It's just important to understand that we have these two types of cells, the main messengers, which are the neurons, and the glial cells, which are the helpers. Here is actually a picture from an electron microscope showing you what neurons and glial cells look like in your brain. So you can actually kind of see they're not colored like this in real life. They're dyed on the electron microscope so that you can kind of get a better look at them. But you can see how that green part, which is the neuron, takes up quite a bit of room. It has lots of branching arms, but all around it are the red cells, which are the glial cells. One thing that you can think about with glial cells is that the word glial kind of sounds like glue, and they are kind of the glue that holds the neuronal system together. So they provide help to the neuron, they provide insulation, communication, nutrient support. So they bring nutrients to the neurons, and then they take waste away from the neurons. Anything we do creates waste. So same thing with our neurons. And because of that, there's way, way, way more glial cells than there are actually neurons in our brain. Okay, so that's what the glial cells do. On the other hand, you have your neurons, and these are, as I put on the slide here, they're the interconnected information processors. Okay, so they're this like web of linked items that pass messages back and forth across your brain and throughout your body. Okay. The nervous system is basically run by these neurons, and each neuron has specific parts. So it has a specific anatomy that we're going to learn about. And the outer surface of that neuron is made up of what's called a semi-permeable membrane. So semi-permeable means that some stuff can permeate or pass through it and other things can't. Okay, so that membrane, that skin that's on the outside of the neuron is semi-permeable, and that membrane allows smaller molecules and molecules without an electric charge to pass through it and stops larger or highly charged molecules. And because it does this, that's actually how we can get the neuron to pass that information back and forth across the web so that it can send those messengers. Okay, so let's look a little bit closer at the anatomy of the neuron. So, neuronal communication is fundamentally like if you think about passing notes between classmates, like if somebody at the front of the class wants to pass a note way to somebody at the back of the class, then they write the note and then they hand it back to the person behind them, and then the person behind them takes it and hands it off to the person behind them, and so on until it gets to its destination. And that is exactly how our neurons work. So here you can see there's kind of this little faded neuron over here, and there's another little faded neuron here at the bottom. This demonstrates that linking characteristic that neurons have. So when we look at this neuron's anatomy, we see that it has this large area up here, which is called the soma or the cell body. And that is where the, you know, that's where essentially the nucleus of the cell is. So if you remember learning from, eighth grade biology or earlier in your 101, 102 cell biology series, you'll remember that cells essentially have a membrane on the outside. On the inside, they have intracellular fluid and there's a nucleus, the base level. Neurons are like any other cell in that they have a cell membrane, that skin on the outside that we've learned is semi-permeable, and they have that nucleus in the middle. And this whole unit is the cell body. Now, coming off of the cell body, you'll see dendrites, which reach out. You can see, think of them kind of arms. The word dendrite comes from the Greek root dender, meaning tree. So you can think about these little branching dendrites as being tree branches that are reaching out. And what are they reaching out for? Well, they're reaching out to be near the terminal buttons of the prior neuron. So let's look at this current neuron. We're going to skip this part right here and go all the way down to the bottom. And when you get to the bottom of this cell, this neuron, you have these terminal buttons. So they're kind of like the opposite of the dendrites. Rather than reaching out to get information, they are reaching forward to send information. At the bottom, these little nubs are called terminal buttons, and they contain what are known as the synaptic vesicles. So you can think of the synaptic vesicles as like little storage houses inside of the terminal buttons that contain neurotransmitters. And if the neurons are the persons that are passing the note back, the neurotransmitters are the note that's being passed. So that's what's being sent from the terminal buttons and what's being uptaken from the dendrites. Now, this middle part that we skipped over is called an axon. And the axon is this long kind of tail that allows that terminal part of the neuron to stretch out and get close to the dendrites of the receiving neuron. Okay, so that's how those messages are passed from neuron to neuron. So let's look a little bit closer at this axon because it plays a special role. Okay, so axons actually vary in length. They can be a fraction of an inch to several feet long, believe it or not. And in some axons, glial cells, remember those helper cells, are going to form a fatty substance around the outside of that axon. That's called a myelin sheath. And what it does is it coats that axon with an insulator. And that insulator is going to allow the chemical messages, the electric messages that are being sent through the length of the axon to go very quickly and easily from one spot to the next. Okay. It may not be continuous, the myelin sheath. There may be small gaps that occur along the length, but the point is that it covers that axon to allow that message to be sent normally from neuron to neuron. If we don't have any myelin sheath at all on our axons, it can be really detrimental. There are actually a couple of different health conditions that a person can have when they don't have fully functional myelin sheaths along the length of their axon. One of them is multiple sclerosis. That is an autoimmune disorder. It basically causes the loss of your myelin sheaths on a large scale throughout your nervous system. And so what happens is that there's interference between the two axons that might be near each other, axons that are kind of sending messages across. There's a lot of interference. And so it reduces the quickness and accuracy with which that message can be sent. And so what that means is that you end up with like dizziness, fatigue, loss of motor control. There can be other types of dysfunction throughout the body. And so while there are treatments that might help a little bit with multiple sclerosis, we currently don't have a cure for that disorder, and it is dramatically affects people's lives. So it's important to remember, even this very small aspect of the way that our neurons work can have large reaching implications if it's not functioning well in our bodies. So let me just show you a quick example of what I mean by the way that the myelin sheath is going to provide that insulation. So you can think about a plug in your house, like any electrical device that you have in your house. Um, they all have on their cord a covering along the length of the cord. You wouldn't want the wires inside of the cord for your electric device to just be out there with no insulation whatsoever. You would want there to be some kind of covering because there are lots of bad things that can happen if that covering is missing. It may send the signal, but it might be sporadic. It may cause interference with other things. So if that naked wire is touching something, it might cause a fire because it's not insulated. So here in this image, you see an insulated normal neuron that's sending a message from that cell body down to the terminal buttons, and it's going just fine and normal. But on the bottom one, you can actually see that the signal is dissipating before it's reaching the end, and that's because of the lack of insulation. So those myelin sheaths are very, very important to protect the signal and make it possible for it to transmit down the length of the axon. All right, so let's zoom in a little closer even still. So now we are looking at what's known as the synapse. Okay, and I want you to think of the synapse as this little spot right here between where that terminal button is very close to the dendrites of the next neuron. Okay, so if you think about that, you can see they're super close, but they're not quite touching. There's a little gap between the sending neuron and the receiving neuron. Okay. And so that little gap between those two neurons is the site of the communication between them. And those synaptic vesicles that we talked about, those little houses that contain the neurotransmitters, can be signaled by that electric message that transmits down the axon to open up and release those specific neurotransmitters into the synapse, into that. It's also called the synaptic cleft, this little space between two neurons. And when the neurotransmitters are released into the synapse, they're taken up by the receiving neurotransmitter using little receptors. Okay, and those are proteins on the cell surface of that receiving neuron. Okay. And because they vary in shape, they can only take in matching neurotransmitters. So the sending neuron has to send the correct neurotransmitters, and the receiving neuron has to receive the correct ones for the communication to be complete. So we're going to zoom in a little bit even closer and have a look at what that specific receptor molecule looks like. Okay, so here now we're even closer in that synaptic gap, and you can see these little molecules that are on the postsynaptic membrane. Okay, the postsynaptic membrane is the receiver, okay, the receiving neuron, and the presynaptic membrane is the sending neuron. So it's pre-synapse, before the synapse. After the synapse, post-synapse is the receiving cell. So those transmitter molecules will only affect the postsynaptic membrane if their shape fits the shape of the specific receptor molecules that are in that membrane. And there's all different kinds of molecules, but they have to be open, they have to be available, and they have to be the right shape. And that's what we call a lock and key relationship. So a key doesn't work. If you try to put it into a key, it doesn't work if it's not the right shape. And the same thing is true of neurotransmitters with their receptor molecules. And that also means that if there's already a key in the lock, even if you have a matching key, it won't go in again. The other key has to be removed before you can put another key in. Okay, so this allows our body to be able to moderate the amount of neurotransmitters that it's taking up in that receptor membrane. Okay, so how does this whole process even work, right? It's a question that, we kind of understand, okay, and a message is received, it's sent, it goes down the axon. I understand that part, but where does the message come from? Like, how does the neuron know to send a message to begin with? Okay. And this is a little bit complicated, but it's not too much. And this is not something that you specifically need to know, like all of the molecules involved. But those of you who are interested in chemistry may find this particular part interesting as to how that message gets kicked off. Okay. So the important thing to remember is that within the neuronal membrane, right, that skin, it's it is semi-permeable. Okay, so some things can come in, but not all things. And the neuron itself exists in a fluid environment, right? It's not like dry inside of your brain, it's like wet. There's fluid. And there's fluid on the inside of the cell membrane, which we call intracellular fluid, which is like the cytoplasm, for those of you who are familiar with that terminology. And there is extracellular fluid, which is the fluid on the outside of the cell. So just like if you were in a pool, okay, there is intracellular fluid, which is like the blood and fluids inside your body, and there is extracellular fluid, the pool water that you're sitting in. So your neurons are basically like chilling in the pool all the time, and they have fluid on the inside and fluid on the outside. Okay. So the neuronal membrane, that skin on the outside of the neuron, is actually keeping the fluids separate. That's really, really important because this electrical signal that passes through the neuron depends on that intra and extracellular fluid being electrically different. Okay, so you can see in this image here, this middle part is going to be that membrane, that semi-permeable membrane. And here is the extracellular fluid, and here is the cytoplasm, the intracellular fluid. And you can see that on the outside, the fluid's net charge is positive. So there's some positive and negatively charged items in the extracellular fluid, and those things balance out to on balance be positive, but that's not the case in the cytoplasm. The net charge here is negative. Okay, so those two fluids have to be electrically different. Okay, and the difference in the charge between these two fluids across the membrane is known as the membrane potential. And that provides the energy for the signal that the neuron is going to pass down the axon. Okay. So these electrically charged ions, these molecules, are dissolved in the fluids. And because of that semi-permeable nature of the neuronal membrane, some of the molecules can get through and some of them can't. So because of that, some types of charged particles tend to become more concentrated on either the inside or the outside of the cell. Okay, so all the time we have our cells sitting in what's called resting potential. So think about a rubber band. If you take a rubber band and you stretch it all the way out as far as it could go, okay, it's not doing anything yet, right? If you stretch this rubber band all the way out, it's not doing anything yet, but it sure has the potential to do something. There's a lot of energy stored in that stretch that if I let one part of it go, bam, it's going to snap. And that's the same thing that's happening because of the electrically charged difference in the membrane potential. Okay, so the ions start to actually line up, as you can see here. This is a sodium ion over here. They're starting to line up, and here's a potassium ion on here on the other side. They're starting to line up along that cell membrane on either side, ready to rush in as soon as the neuron goes active and the membrane opens up its gates. Okay, so that's what this other big thing in the middle of the cell membrane is. That is a sodium potassium pump that allows the movement of ions across the membrane. So this is how we mean when we're saying it's semi-permeable. Sometimes the cell allows things to pass through and sometimes it doesn't, okay? And that sodium potassium pump right there is a gate that will allow these ions to move once it opens up. Okay, so when the ions are in a high concentration area, they're ready to move to a low concentration area, and positive ions are ready to move to negatively charged areas. Okay, so remember, everything with your body, everything that you're going to learn in biology and chemistry, the base thing you need to remember is that every element and molecule and particle wants to be stable. Okay, we want to be stable in a stable situation. The rubber band does not want to be stretched out. It wants to be just laying there doing nothing. No, everything's stable, nothing's about ready to change, okay? But because these ions build up, they start to develop more and more difference, and that becomes unstable. So they want to go move into the other area where they'll be more stable. Okay, if it's too positive, they want to go where it's negative. Okay, if it's too concentrated, they want to go where it's less concentrated to balance it. And they want to try to get there so that they can maintain that homeostasis. Not too much, not too little, just balanced. Okay, so in this resting state, right, we tend to have sodium at a higher concentration outside the cell, and it will want to move into the cell where there's not as much sodium. Same thing with potassium. Potassium on the other side, it's getting too concentrated. It wants to move outside of the cell where there's less potassium. Okay. So the inside of the cell is slightly negatively charged and the outside of the cell is slightly positively charged. That pushes the sodium a little bit more strongly as well, trying to get it into the cell. Again, everything's just trying to find a place where it can be more balanced. If there's too much, it wants to move where there's less. If it's too negative, it wants to move where it's positive. Okay. So when that happens, when it reaches a critical point where the charge differences are too high, where the concentration differences are too high, then that will trigger that sodium potassium pump to open and allow the exchange of some of these ions. Okay. And this is what that looks like. So here you can see, oh, it got too high. Everything wants to change places. And once that happens, you can see how that pushes that information down the whole length of the axon. Okay. So whenever that change happens, whenever the tension, just like we're pulling the rubber band, whenever the tension gets too much, eventually it's going to snap and it's going to move really rapidly, quickly, and then things will start to build up and become concentrated again. And that's how we create the action potential, which is that movement down the length of the axon. Okay. So the axon, the action potential is created by that difference in charge, and that's what moves that messenger all the way down the end of the axon. Hopefully that makes sense. Like I said, you don't have to remember all the specific like little chemistry pieces to that. Mostly it's important just to understand that there's a change in charge, and that's what causes that to happen. Okay. So once that does happen, okay, once we move from that resting potential state and the sodium potassium gates open up, and then all of those sodium positive ions and that are propelled by the concentration differences and by the charge differences to move into the cell, that internal charge becomes more positive and that pushes things down. Okay, that process is actually called depolarization, and we'll talk about that a little bit more in just a minute. But once that has happened, you can see what's exactly occurring in this synaptic cleft. So here, before this happens, you can see all of the neurotransmitters are inside of the synaptic vesicles. Okay, nothing is happening in here. There's a calcium channel here that's, gonna do some stuff potentially, but for the most part, there's nothing going on inside of this synaptic cleft. But once that charge happens, the action potential is pushed down the length of the axon to the terminal buttons that influences those synaptic vesicles to open up to send that neurotransmitter inside of the synaptic cleft where it's brought in, it's taken up by those receptor molecules, by those cells down here, at the very bottom of that receptor cell. And then anything that's left over is taken back up into the original sending neuron. So if there's too much, it just takes it right back up with this reuptake molecule, and that prevents it from having to remake neurotransmitters that might so that they go to waste. It just saves you energy and time. Okay. So lots of these additional pores open. All of this stuff moves really rapidly. Okay. And what that looks like if you look at this chart here is that the charge across the membrane changes really dramatically. Okay, and like I said, there's that resting potential. And then once we reach what's called the threshold of excitation, then we, boom, reach that peak action potential really, really fast. And then as we go down, we start to repolarize. So remember, those charges were getting too concentrated, too opposite. So they kicked off that threshold of excitation, which created the action potential. Now they have to go back to normal. So back to normal repolarization. There's a small period of hyperpolarization, but that levels off very quickly. And then we get right back to where we were. Okay. So the important thing to realize from this particular graph right here is this, the threshold of excitation. What does that mean? Well, that means with neurons, we have an all or none system. There is no such thing as a small action potential. Either it happens or it doesn't happen. So we have to have the the minimum threshold to make that action potential happen. Okay, there has to be a sufficient signal. There's no in between. Just like if you send an email or a text message, right? I'm sorry if I'm covering the microphone. You guys know I talk with my hands, so I kind of get all crazy. So I'm sorry if that's distracting. Um, think about if you're sending a text message. Okay, you can think about sending it all you want, but the message is not sent until you hit send. And then once you hit send and it's on the way, you can't stop it, right? It's already sent. There's no such thing as kind of sending a text, right? You didn't send, it's not like a little sending a text. You either sent it or you didn't send it, right? So because it's all or none, the action potential is recreated, or rather what we call it propagated down the length of the axon. So every single point where that, here, I'll show you again that image. At every single point, the full action potential is created. It doesn't lose its strength as it passes along, because in order to trigger that reaction, it has to have reached the minimum threshold for excitation. Okay. So this all or none property actually explains why your brain perceives an injury to your toe as equally painful as one to your nose that's right next to your brain. Okay. If you stub your toe super hard, that's going to hurt just as much as if you bump your nose, really hard. The distance away from your brain is irrelevant, because as long as it meets that minimum threshold for excitation, then it's going to continue at that strength all the way to the brain, from wherever the site of the injury is all the way to the brain. Okay. And then again, provided that you have functional myelin sheaths. Okay. So hopefully that makes sense, as to that all or none property. And you remember that I was talking about how there's that reuptake molecule, and that once we send those neurotransmitters into the synaptic cleft, once certain number of the receiving proteins have taken up the neurotransmitters, then the neurotransmitters that are left in the synaptic cleft are brought back in to that sending molecule, rather that sending neuron, so that it can be reused at another time. And this is just, again, to be, not to be wasteful and to not have to create more neurotransmitters than are needed because it takes your brain a lot of energy to create those. So, because of how that works, because of the way that our neurotransmitters work inside the synapse, we actually are able to exploit that process to create what's called psychoactive drugs, so psychoactive medications. Okay. So psychoactive drugs can correct neurotransmitter imbalances. And because of that, it's important for us to know how those neurotransmitters binding to the receiving neuron, how they work. Okay. So if we use a mimic of a neurotransmitter, it's going to fit inside of those receiving proteins, right, that are on that surface of the receiving molecule, the receiving neuron. And when that happens, it can either make the signal bigger or make it smaller or not at all, or kind of somewhere in the middle. And the words that we use to describe that are agonists and antagonists. Okay. Agonists mimic a transmitter. They mimic a neurotransmitter at the receptor site, and so they make it bigger. So let's say you only have a small amount of a certain type of neurotransmitter, and so the effect that you get from it is usually very small. We can give you an agonist, a medication that mimics that neurotransmitter, and it's going to bind to a lot more of those receptor cells. And then that way, that experience of that neurotransmitter for you is going to be bigger. You're going to experience it more. Okay. On the other hand, we have antagonists. Okay, antagonists block the normal activity of a neurotransmitter at the receptor, so it makes it weaker or not at all. So remember what I said earlier, if you don't have a receptor, if all the receptors are blocked, then you can't put a neurotransmitter into it. And these antagonists can block the receptor without having any effect at all. So they block the receptor and don't allow a neurotransmitter to get in, and that weakens the effect or makes it not at all. It creates no activation. Now, sometimes we don't want there to be no reaction and we don't want there to be full activation either. We just want something kind of in the middle. And in that case, we might use an agonist and an antagonist at the same time. So some of those are going to be filled with agonists, mimics of the neurotransmitter, and some of they're going to be filled with antagonists to block so that we end up with a little bit of a moderated experience. And that's the case if a person has too much of a neurotransmitter, right? And we don't want to block them all. We don't want, or maybe they don't have enough of one at all. We don't want to block them all. We want to help them work, but we don't want it to work too much, right? So we do a little bit of both. Okay, so that's how psychoactive drugs can exploit that neurotransmitter system to create different feelings in you. But we've been talking about neurotransmitters. We've been kind of just discussing them without really talking about what they are specifically and what they might make you experience. So here is a list. It's just a short list of major neurotransmitters and how they affect behavior. Okay, so some of these you will have heard of before. Many of you will know about dopamine and serotonin, right? So the dopamine is involved in mood and sleep and learning. And the way that it feels when we get dopamine is pleasure. So we get more pleasurable feelings. And we might have sort of a suppressed appetite when we experience dopamine. Okay, so dopamine is the pleasure neurotransmitter. Now, serotonin, on the other hand, is responsible for mood and sleep. Okay, so when we have appropriate amounts of serotonin, our mood is modulated. That means it's going to be right in the middle, stable. Okay, it's not going to go up and down crazy. When we have good serotonin, we might also experience a suppressed appetite again. So you can see when we have proper amounts of dopamine and serotonin, we are less likely to say stress eat or eat because we're sad, right? On the other hand, we have certain, we have certain neurotransmitters that are responsible for increasing, appetite, and that those would be like more in your stomach, like ghrelin or leptin. Leptin tells us when we're full. Ghrelin tells us when we're hungry. Okay. Other neurotransmitters, acetylcholine, that one is involved in muscle action and memory. Okay, so when you have acetylcholine, that can get you more focused. It can also help you with your reaction time, acetylcholine. Beta endorphins are responsible for pain and pleasure. Okay, so we can, when we're experiencing pain, beta endorphins are our own body's endogenous morphine, essentially. So it decreases our anxiety, it decreases our tension. It's our own little painkiller. Okay, so people say if you exercise, you feel great afterwards because you get all of the endorphins, and that's true because exercising hurts. Exercising feels kind of bad on your body, and so your body sends you these beta endorphins to help you feel better. Okay. Gamma aminobutyric acid, that's GABA. Okay. That is responsible for, calming down, going to sleep. GABA is an inhibitor. Okay. GABA shuts stuff off. So when your mind is going 100 miles an hour and you can't stop, GABA is what is telling it to stop, slow down, chill out, go to sleep. GABA is very important for that. Okay, glutamate, important for memory and learning. Okay, it enhances our memory, it increases our ability to learn from our experiences. And norepinephrine is another one you guys might have heard of. That's responsible for increased arousal, suppressed appetite. It affects our heart, intestines, our alertness, because that's the one that gets activated when we are in our fight or flight. So when we're in our fight or flight and we need to fight or run away, norepinephrine floods into that synaptic cleft and helps us to get moving. Okay. So, this is just some of the major neurotransmitters and some of the ways that they may affect behavior. And sometimes we can kind of start to see how, maybe we might have a problem with one neurotransmitter, how that might create some problems in our behavior. So, one way that we can fix that, for example, with serotonin, is using reuptake absorbing blockers. So, if anyone has ever heard of an SSRI, that's an antidepressant medication. SSRI stands for Selective Serotonin Reuptake Inhibitor. So now you know exactly what that means, right? Because you know about the reuptake, molecules on that sending neuron. When serotonin floods into the synaptic cleft, we think that for some people with depression, it may be the case that the serotonin is taken back up too quickly and it doesn't have a chance to fill those receptor sites for enough or long enough to keep your mood stable. Okay, so what we want to do is we want to make sure that those synapses stay full of serotonin to keep your mood right in the middle. And so we block one of the reuptake molecules to keep more serotonin in the synaptic cleft for longer. Now, in full transparency, there's been some research that has come out that has questioned whether or not this is an effective treatment for depression. But this is one idea for a way that it might help in people with depression who have chronic low mood. Okay. Another case that is pretty timely for kind of the experience that we're going through right now as a society, is another type of psychotropic medication that works by blocking, not only blocking, but kicking out certain types of received messengers, neurotransmitters, in this case, artificial neurotransmitters. So I'm going to show you a video about this particular drug. And while you're watching it, just think if you recognize this, have you ever heard of this before? And just think if this is something that you also now understand a little better because you know what's going on at the brain level.
Naloxone is a medicine that rapidly reverses an opioid poisoning. It's an opioid antagonist. That means that the naloxone molecule attaches to the same receptors in your brain that otherwise could be a receptacle for opioid molecules. Naloxone acts like a barrier preventing opioids from having an effect. But more than that, naloxone will actually kick out opioid molecules already activating neurons and causing problems. So that means it will rapidly reverse the effects of opioid poisoning on the body.
Okay, so there you can see the drug that I was talking about was naloxone. You can see how it's an antagonist, and even more so, it can rapidly kick out those opioid molecules from those receptor cells. And what that means is that if a person is overdosing on opioids, we can apply naloxone we can actually save their life. So this is something that is a life-saving medication. They tend to be unhappy with you when you do that, but that's a part of, the deeper condition of substance use disorder that is also, a crisis right now. But at least we can stop people from dying of opioid overdoses by having this in our first aid kit, or, if we work with people who have a higher risk or propensity for opioid overdoses, having those on our person. So that's just something to think about, how we're using this process that's going on in our brain to be able to help people. Okay, so now that we understand the basics, right, we know exactly how that neuronal communication process is working. Now we're going to talk about the bigger systems of our body. So we understand neuronal communication. How do neurons and hormones, we're going to talk about as well, communicate across our whole body? So how is the brain connected to the rest of us, I should say? Okay, so the nervous system is made up of two parts. Okay, you have the central nervous system, the CNS, and the peripheral nervous system, the PNS. Okay, the central nervous system is just the brain and the spinal cord, and the peripheral nervous system actually has two parts to it. So our nervous system has two parts, and one of those parts has two extra parts. Don't get confused. I'll walk you through every part of it, but just so you know. The two subparts of the peripheral nervous system are the somatic and autonomic nervous systems. Okay, so some people find it easier to see an image of them kind of separated. So here you have the central nervous system. This is the brain and the spinal cord. That's it. That's all the CNS is. And then on the other side, we have the peripheral nervous system, which is everything else. Okay. So let's begin by starting with the central nervous system, the easy part, and then we'll move on from there. So the central nervous system, the brain, contains approximately 100 billion neurons and 100 trillion synapses. That's a lot. That's crazy. Like, just think about that for a second. That's insane. In everyone's mind, in everyone's brain, 100 billion neurons and 100 trillion of those little synapses, and they're working all the time to keep you going. Okay, so the average weight for a brain is about 3 pounds for a male and about 2 and 3/4 pounds for a female. Really, the averages are also based on body size. So males tend to be a little bit bigger than females, and weigh a little bit more. And so that's why the disparity is there. The spinal cord is about 18 inches long, so a foot and a half, and it contains about 1 billion neurons. Okay, so your brain has 100 billion neurons, and your spinal cord actually also has about 1 billion neurons. A billion with a B. Okay, we're going to talk about how your spine uses its neurons in a minute, but for now, let's leave the central nervous system and move into that peripheral nervous system. So the peripheral nervous system is made up again of those two parts, the somatic nervous system and the autonomic nervous system. And actually, funnily enough, the autonomic nervous system is also made up of two parts. I don't know. I didn't invent it. That's just how it is. Each part has two parts, and one of the parts is made up of two more parts. But I promise it ends there. Okay. So you start with the somatic nervous system. That's for our sensory and motor signals to and from the central nervous system. And then the autonomic nervous system controls the function of our organs and glands and has the sympathetic and parasympathetic nervous systems within. Okay, so sympathetic is what prepares us for fight or flight, and parasympathetic is rest and digest. It calms us down afterwards. Okay. So this image kind of can help as well to see how things are sort of broken down. So you have your central nervous system, the brain and spinal cord, and then you have your peripheral nervous system. In the peripheral nervous system, you have the autonomic and the somatic system. Okay. And then in the autonomic system, you have the parasympathetic and the sympathetic system. So sometimes this can help just to think about all the ways that it's kind of broken down. It's not, I mean, it's just what it is. Like, you have to kind of memorize it, but it's going to kind of help you to picture it big picture. Okay. So we're going to go through each one of those peripheral nervous system parts in detail. So we're going to start with the somatic nervous system. Okay. So the somatic nervous system is associated with the activities that we traditionally think of as conscious or voluntary. Okay. So the relay of sensory and motor information. Okay. So conscious voluntary body movements. You can think of them as being just things that you do. All right. So when you're, lifting weights to exercise your bicep, that's in the somatic nervous system. If you're hearing music, that's in the somatic nervous system. Kicking a ball, seeing the sky. So anytime your skeletal muscle actions are involved, or if you're receiving sensory input, so sights, hearing, smells, all that stuff, that's all the somatic nervous system. Okay. And the somatic nervous system has a couple different types of neurons within it that allow those things to work. So you have your sensory, your afferent nerves, and your motor or your efferent nerves or neurons, same thing. Okay. So your sensory nerves are the things that are coming in from outside in the world up to your central nervous system. So feelings I have on my skin travel through the afferent nerves, sensory nerves, up to my brain and tell me what sense I'm feeling. Motor or efferent nerves are things that are coming from my central nervous system, commanding my muscles and skeleton to do stuff. So if I want to kick a ball, then my brain decides I want to do that, and motor or efferent neurons are sending that signal to my leg to make me do it. Okay. I did not name these nerves. Okay, I that was not my doing. I think it's just to confuse you. I have no idea. Those two words sound exactly the same, afferent, efferent. The way they're acting like that's just a normal way to name them is crazy. That's just my opinion, but there's a mnemonic that can help you remember, and it's same. So because those two words sound the same, you can remember which one is which by thinking of the word same. S-A-M-E. S-A, sensory is afferent. M-E, motor is efferent. So that's what helps me, but oh my goodness, like this is I don't know why they have to make it so tricky, but that's just the way that it is. Okay. So you can just think about each nerve is this bundle of neurons forming a two-way superhighway. Okay, thousands of axons. All right, both afferent and efferent. The afferent ones are coming in to the central nervous system. The efferent ones are going out to your muscles and skeleton. Okay. So you say to me, all right, that's my voluntary motor system. You said that's how I can make things move in my body and how, I can experience sensations and stuff. But what about my non-voluntary or my reflex system? Well, that is also controlled by sensory and motor neurons, but they're actually mostly for those non-conscious decisions are going to be actually processed by your spine. Okay, so the somatic reflex arc involves sensor neurons. So I see an apple, and then my sensory neurons are sending that to my brain. And then I'm making the conscious thought that I would like to pick up the apple and eat it. So my brain sends that thought through my motor neurons to my arm, which causes me to pick up and eat the apple. Okay, however, if I have a reflex, that's not going to my brain. My brain is not involved in a reflex. Okay, so when something is hot and I touch it, like I touch the stove and it's hot, my hand is pulled away through an automatic reflex initiated by my spine, not by my brain. Okay. In survival, the spinal reflexes allow the body to react extraordinarily fast because even though our neurons are super fast and they can get that information to the brain super fast, sometimes your hand is cooking on the stove and we don't have time to wait. So it just has to go through the spine where there's those billion neurons that we talked about and get your hand away as quick as possible. So here's what that spinal reflex arc looks like. So we hit the knee, right where you have that involuntary nerve. Your doctor will do that. You have that little kick reflex. Okay. And that will go up the sensory nerves into your spinal cord, not your brain. Your spinal cord will then initiate your motor nerve to respond with the kicking reflex. Okay. So I don't know if I'm repeating this too much, I'm sorry, but I remember when I first learned this, I was like, my mind was, I was that galaxy brain, like moment when my brain was absolutely blown by this, the reality that my sensory reflexes, my reflexes are not involving my brain at all. So that's crazy. I hope that some of you are also similarly blown away by that. Because I'm feeling a little self-conscious. Maybe I'm just dumb, but I thought that was so cool when I first learned that. Okay. So that's your sensory motor system. Okay. Now let's talk about the autonomic nervous system. So this is that one that also has two parts to it. Okay, so the autonomic nervous system, you have the parasympathetic nervous system and the sympathetic nervous system, and they work together by causing fight or flight and then allowing you to relax after fight or flight. Okay, so when you're presented with some type of danger, some type of threat, your sympathetic nervous system is engaged, and that causes your pupils to dilate to let in as much light as possible. It inhibits salivation, so you start to get sort of dry mouth. Your heart starts beating, your bronchi become dilated, so it kind of feels like I'm getting so much air. You breathe heavier. Your digestion is actually inhibited, so you may start to feel like, oh, I have a sick stomach. Anybody who gets like stress constipation, that's probably from the sympathetic nervous system. And it also inhibits the contraction of the bladder, which is why sometimes when people are very frightened or startled, they can, wet their pants because the bladder just will let go because you don't need to be holding on to any weight if you're running away from a tiger. Okay. Now, once you're away from the threat or the threat has been mitigated, then your parasympathetic nervous system is what's going to kick in, and that's going to constrict the pupils back. Okay. We don't need our eyes to be that open wide, get our salivation back, slow down the heart rate, constrict the bronchi. We don't need to have our lungs like constantly taking in like 1,000 pounds of air. Stimulates digestion. So if anyone's ever been through an experience where they were very stressed and then finally the stress thing ended, then you felt super like hungry and you were ready to eat. Digestion is stimulated and it causes the bladder to contract so that you're not peeing yourself after the threat is gone. Okay. So again, remember in your body, as with all things, you're trying to maintain homeostasis, that state of balance, of equilibrium, where our conditions are just maintained at optimal levels. Okay, so when you're in fight or flight, naturally your body and your brain will lean towards rest and digest, ready to go back to normal. So always when you're in one system, the other system is ready to activate when needed. Okay. You can't be in one all the time. You can't just be in a state of fight or flight all the time. That's really terrible, terrible for your body. But you also can't be in rest and digest all the time because sometimes you need to do stuff, right? Like we have, sometimes we need a little adrenaline to start, doing to meet a challenge. Okay. So both of these things are necessary and they both want to be maintained in balance. All right, so that is the CNS and the PNS. So that's our nervous system. Now we're going to talk about the endocrine system, okay? And while the endocrine system, while the nervous system was the same for everybody, the endocrine system is going to vary depending on your sex. Okay, so males and females have different glands and hormones. So most of it's the same, but there's a couple areas where that's different. So there's going to be different, slight differences between males and females on this one. Okay, so the endocrine system consists of a series of glands throughout our body, and they produce a particular type of substance known as hormones. Okay. Hormones also regulate behaviors. Just like our nervous system sends electrical signals to change our behavior, our hormonal system also regulates behaviors like aggression, mating, parenting, eating, all kinds of things. Okay, so hormones influence our behavior, but sometimes our behavior actually influences our hormones. So things that we're doing in our life can cause certain hormones to be secreted as well. Um, any any type of research into the way that our endocrine system impacts our psychology, so our behavior and our mind, is in the field of behavioral endocrinology. Okay, so it's a really interesting emerging field. All right, so let's start with the basics, the parts of the endocrine system. And then this single being will have both the female and the male glands, even though we don't see that in nature. There's this just for simplicity of the diagram. So we start with the pineal gland. Okay, the pineal gland is towards the rear of the brain, and the pituitary gland is towards the front of the brain. They're both kind of in the midbrain region next to the hypothalamus and subordinate to the thalamus. And what we see the pineal gland doing is that it regulates melatonin. So it's really about our biological clock and kind of knowing when to wake up and when to go to sleep. Okay. The pituitary gland, we call that the master hormone, or excuse me, the master gland, because it releases all of the hormones that are related to our ovaries, kidneys, uterus, breast tissue. Many, many organs are controlled by the pituitary gland. It's also the one that kind of kicks off our puberty. So it's a very important gland. The next one is the thyroid gland, which is near the thyroid cartilage of the larynx in the throat. Okay, that one is responsible for thyroxine and controls a bit of our liver. It has to do with our metabolic rate. Okay, so important there. Then we have the thymus. The thymus is underneath of that. Okay. We have beneath that adrenal glands and the pancreas. Okay. Adrenal glands are going to be responsible for our adrenaline system, cortisol, stress, fight or flight. Pancreas is responsible for our insulin and glucagon. So that has to do with eating and appetite. And then for females, we have the uterus and the ovaries that are going to produce estrogens like progesterone. And then in males, we have the testes, which are going to produce testosterone, androgens like testosterone. Okay. So let's go through each one of those major glands in a little bit more detail. Okay. The pineal gland, like I mentioned, responsible for that melatonin secretion. Okay, that helps us to know what time it is. Is it time to go to sleep? Is it time to wake up? It kind of keeps our biological clock in order. Okay. The pituitary gland, like I said, that descends from the hypothalamus at the base of the brain. Okay. And it really acts in close association with it. Okay. The hypothalamus being that master switchboard where it's like sending information to all the other parts of the brain. Okay. It basically messengers hormones. This, right, we call the pituitary gland the master gland. It's a messenger hormones control all of the other glands in the endocrine system. Okay, it mostly carries out instructions from the hypothalamus, but it secretes growth hormone, endorphins for pain relief, a number of super key hormones that regulate all kinds of fluids levels in the body. So the pituitary gland is extremely important. And a lot of times we associate it with our sexual maturity. So going through puberty, having a menstrual cycle, growth and division. When you go into labor, pituitary gland is responsible for birth contractions, milk production, just every single part of your body can be linked back to the pituitary gland in many ways. Okay. The thyroid gland in the neck is going to regulate growth, metabolism, appetite. So if you've ever heard of a condition called hypothyroidism, that's when an individual has a poorly functioning thyroid gland. It doesn't produce as much hormone as it needs to. And so that person will need to take medication to replace the thyroxine in the body. Okay. You can also have hyperthyroidism where you're producing too much of that thyroxine, and that can cause agitation, difficulty sleeping, bulging eyes, weight loss, difficulty putting on weight. Okay. So you think about it as associated with our metabolic rate. So how much energy we get through in a day on our own. All right. The next thing is the adrenal glands, and those are kind of like little kidney beans. They sit on top of your kidneys and they are responsible for that stress response. Okay, so they send out epinephrine, they send out noradrenaline. Okay, they send out cortisol is responsible there as well. So to get you amped up when you need to be stressed, when you need to be responding to a threat. Okay, The pancreas is nearby, a little bit underneath by the kidney, and that is going to be secreting hormones that regulate our blood sugar levels. So if you have a problem with your pancreas with those hormones, then you might have a condition called diabetes. Okay. People with diabetes don't produce enough insulin, so they have to take medications that stimulate or replace insulin production. And then they also have to control the amount of sugars and carbohydrates they consume because the pancreas is supposed to respond to sugar in the bloodstream. Okay. So when you have problems with diabetes, then that can make that function not work. Okay. You can get too high blood sugar, which can cause all kinds of problems. Okay. And then finally, the ovaries and the testes, like I mentioned, we call those the gonads collectively. Okay. And they're going to secrete those sexual hormones. So estrogens like progesterone, like prolactin. Okay. And then androgens like testosterone. So the king estrogen is progesterone and the king androgen is testosterone. Okay. And they're responsible for things like menstrual cycle. They're responsible for different characteristics in the body. So male and female characteristics that we see at sexual maturity. Okay. So now I mentioned earlier that our hormones control our behaviors, and I've explained many ways that they do that, but also our behaviors can impact our hormones as well. Okay, so because, if we have an excretion of estrogens in a female, we see female sexual behavior. We have excretion of androgens in the male, we see male sexual behavior. Okay, when our pancreas secretes insulin, we, are able to take up food. Okay, when our that tells us that we're hungry when our pancreas is secreting insulin, it's preparing for food to come in. Okay, when our adrenal glands are secreting epinephrine, we start to feel amped up, like there might be a threat around. Okay, when our posterior pituitary gland is starting to secrete oxytocin, we start to have bonding parental behaviors, right? So the hormones can control the behaviors, but also the behaviors can affect the hormones. So let me ask you guys a question. Have you ever been like going out with some friends and they were like, "Hey, we're gonna go to the mall, maybe get something to eat." And you're like, "Yeah, I'll come along with you. I'm not really hungry, but I'll just hang out with you guys when you get something from the food court, no problem." And you're not hungry, and then you go there and you're walking around and then you go to the food court and you smell the food, and then you're like, "Dang." And then you start getting your mouth starts to kind of water, and then you're like, actually, I am hungry now." That is behavior affecting hormone release. You went where the food was and you smelled food, and then your brain was like, sounds like that sounds really good. And then your body was like, "Oh, okay, crap. Let me produce some insulin so that you can be ready to eat." And then you're hungry. Okay. Another example for those of you who have had children before and have breastfed, if you hear a baby crying, sometimes that can cause an oxytocin release in your brain that causes your milk to let down, and then your breast can leak milk when your baby is not even ready to eat. You just hear crying, and so that experience causes the hormones to activate. Okay, so they're both one side or the other can definitely, the hormones can affect behavior, but also our behaviors can affect hormone release. Okay. So one thing I want to talk about is a common question that we look at is, are toy preferences learned or could they be influenced by hormones? Okay, so right now, science has what we call a primacy of learning as an explanation for toy preferences, which means that little boys play with quote unquote boy toys because they're taught that those are toys for boys, and little girls play with quote unquote girl toys because they're taught that those are toys for girls. Okay. And so they take in that experience from their learning environment, and so that's why they go one way or the other. However, there has been some research that has looked in monkeys, for example, to see whether or not they are preferring girl or boy typical toys. So they divided the girl typical toys as dolls and like mini kitchens and things like that, and male, typical toys, boy typical toys as cars or balls. Okay, things that move tend to be preferred by boys. Okay, and it turns out that for these vervet and rhesus monkeys who are not brought up monkey doesn't have a concept of boy or girl toys. They're monkeys, right? And so because they don't have that concept, it seems interesting that vervet and rhesus monkeys would show sex segregated toy preferences, and yet they do. Okay. Then looking again to see whether or not there's some aspect of learning in human children, we see sex differences in toy preferences emerge by 12 or 24 months, and even earlier in life. Pre-verbal infants, as young as 3 months old, were assessed, and what they do is that they show them pictures. So little tiny infant babies can't like reach for things, but they do hold eye gaze on things that they like. So we know, for example, that babies like faces because they look at faces longer than anything else. They love looking at their mom's face, especially. Okay, so when we see what attention grabbing, toys are there for boys and girls, we actually see that as little as 3 months old have sex differences in toy preferences. So girls preferred dolls while boys preferred trucks. And obviously, this doesn't prove it, but it suggests that there's something going on prior to learning because they're only 3 months old. So, I guess they may have had some exposure to expectations, but they don't really have a conscious understanding of what those expectations might be. And yet they still had a preference. So, the last time or the last type of experiment that was used to look at toy preferences was with girls with a condition known as congenital adrenal hypoplasia. Okay, and congenital adrenal hypoplasia only exists in female children, in females, and we see that they have adrenal glands producing more androgens. Okay, so girls with congenital adrenal hypoplasia produces more androgens than a girl without that condition. Okay, and what we found was that girls who have this condition prefer masculine toys more than girls without the condition. So they also found that there was a dose response relationship, which means that um, if the girl was exposed to more androgens as a fetus, then they tended to be more interested in masculinized play behavior. Okay. So they saw that having more testosterone encourages people. I mean, maybe it doesn't encourage it, but having more testosterone is associated with, remember what we talked about last week with experiments versus correlational research, right? This is just correlational, but they found that girls that have higher amounts of androgens in their body tend to be more interested in quote unquote boy toys than girls that don't have those androgens in their body. So obviously we're not together, so we can't have a big discussion together, although I really love it when we have this discussion topic. But one thing I would love you to think about on your own is, oh, I haven't changed the slide. Well, the real discussion question that I wanted you to think about, ignore what's on this slide. The real discussion question that I wanted you to think about is whether you feel that the hormones that are in a person's body may also influence their preferences. So, do you think that liking quote unquote girl toys or quote unquote boy toys is just as a result of society telling us what we're supposed to be allowed to play with? Or do you think hormones play a role? Again, this is all up in the air. I don't take an opinion. I just share with you what the data is showing right now. So I would love to have you think about that and just come to your own conclusions. And if you're interested, I can also share some of the research with you. Okay, so just to summarize this first part of the lecture, we talked about the basic structures of the neuron and the function of each structure. We talked about how messages travel through the neuron, how psychotropic medications work, the role of the nervous system and endocrine systems, that fight or flight system, and how hormones can also impact behavior. Okay? So, as I said, hopefully you can use a slide like this to help you prepare in your study plan for your exam. Okay, so we're going to move into the next section. I encourage you, take a break if you'd like to take a break. I know it's a lot. Normally in class we would take a 15-minute break or so. So take a few minutes, check over your notes, pause the video, come back to it another time, or come right back to it. And we're going to go into the second half of the biopsychology unit where we're talking about parts of the brain and nature versus nurture. Okay, so Welcome back if you took a break, and good for you for taking a rest. And if you didn't take a break, let's just carry on. Okay, so we start with parts of the brain. We're going to talk about the two hemispheres of the brain and the location and function of the lobes. Then we're also going to talk about the limbic system, midbrain, and hindbrain, and brain imaging and what different types of imaging that we have. Then at the very end, we're going to do a little bit about nature and nurture. So, the history of that question, the principles of evolution, genetic variation, and mutation, and epigenetics. That's a really small little bit just to think about how, yes, we have all of these aspects of our brain that maybe make certain behaviors or attributes more or less likely, but how the environment can also impact things as well. Okay, so this section, we're going to talk about the specific parts of the brain and their roles and functions. Okay, this is not an anatomy class, but you'll see how important it is to kind of understand the parts of the brain and what they do so that we can later begin to understand more about the mental processes and behaviors that we're going to talk about later in the term. Okay, so to give you kind of an overview, okay, the brain is an incredibly complex organ. It's comprised of billions of interconnected neurons and glia. Okay, you can see here, this is kind of a breakdown of some of the parts of the brain. You are not going to have to remember all these numbers and all these other things. I'm just kind of showing you, we have a lot of specific things that we have identified about the brain and how it works. And we're going to kind of go into this localized experience of the brain, what these parts do and what they're associated with. Okay, step by step. So important to know, the surface of your brain is called the cerebral cortex. Okay, it's very uneven and it's really bumpy and lumpy. It has all these folds and bumps. Okay, the folds are known as gyri, or the singular gyrus. So one fold would be a gyrus. Okay. Um, and excuse me, the little creases are called sulci. Okay, they are the grooves in the brain. Okay, and the bumped up parts are the gyri. Okay, so you have one gyrus, multiple gyri. Those are the bumps, and the grooves are the sulci. One would be called a sulcus. Okay, so you can see here in the picture, it's kind of generally pointing to the brain, right? There are the grooves that are called the sulci and the bumps that are called the gyri. And then down the whole center of the brain, you have the most prominent sulcus or groove known as the longitudinal fissure. And that is what divides our brain into two halves, which we call hemispheres, the left hemisphere and the right hemisphere. Okay, so the important thing to know about these two hemispheres is that our brain has a process called cross lateralization. Okay, and what that means is, just to be confusing, just to make things even harder, is that each hemisphere actually controls the opposite side of the body. Okay, so my right hemisphere of my brain is going to control my left side body, and my left hemisphere of my brain is going to control my right side body. And we normally have kind of a complete copy of things on each side of our brain, but there are some specializations in function, especially in language. Okay, language is a one-sided thing. So when you think about anything being one-sided, seems like it doesn't make sense, because then you'd have only one side of your brain able to do something, only one side of your body able to do something. That's why we have this really thick band of neural fibers in that major sulcus, that big crease in the middle that separates our brain into two halves. That's called the corpus callosum. Okay, and the corpus callosum connects our left hemisphere to our right hemisphere so that both sides can talk to each other. Okay, here you can see in the bottom, there's an actual dissected brain here. And you can see that white band of fibers, the corpus callosum, there in the center. Okay. So the left hemisphere has been shown to be superior at forming associations in memory, selective attention, and positive emotions. The right hemisphere has been shown to be superior in pitch perception, arousal, and negative emotions. Okay. So there's some areas where the brain is better at one thing on one side than it is on the other side, but the corpus callosum allows those two sides to talk. Now, normally, we're not really aware of the role that our two hemispheres play in our day-to-day life. Okay. We just go about our day, and if one thing is one side of our brain or the other side, we would never even know. We never think about it because of the corpus callosum. However, there are some people for whom we have had a look at what that might look like for them. Okay. And what that comes from is that there is a cure for extremely severe epilepsy. And if a person has extremely severe epilepsy where we can't control their seizures and the seizures are spreading and they're getting worse, then doctors can sever the corpus callosum. So it can cut it to prevent the seizures. And it's an extremely effective treatment option for those patients for whom the epilepsy is serious enough. However, it results in individuals who have what's called split brains. Okay. So after the surgery, these split brain patients show all kinds of very interesting, unusual behaviors because of the way that our brain is both cross-lateralized and also has some hemispheric specialization. Okay, so for example, a split brain patient is not able to name a picture that is shown in their left visual field. Okay, so I put a wall between your eyes so that this eye can't see what's going on over here and this eye can't see what's going on over there. And I show a picture just to the left eye. Okay, so which side of the brain is seeing the picture? Think about it yourself. Okay, the side of the brain that's seeing the picture through the left eye is the right side of the brain. And the right side of our brain is largely nonverbal. It can't really talk very well. The verbal side of our brain is our left hemisphere. So if we ask them to recreate the picture with their left hand, which is also controlled by the right hemisphere, they can draw or write the name of the picture, but they can't say it because of the way that the other side of the brain controls our verbal ability. Okay, so when the more verbal left hemisphere sees the picture, okay, even the picture that the hand drew, okay, so let's say I show a picture of a banana to a split brain patient's left eye. Okay, the right brain can't communicate what it saw, but it does know what it saw. So it takes the left hand and it draws a picture of the banana. And then I show the picture of the banana to the right eye, so the verbal left hemisphere, and the person's able to say, "Oh, it's a banana." Interestingly, though, if you ask the person, "Why did you draw a banana?" they won't be able to know because the hemispheres of the brain are separated. So the right brain can't tell the left brain, "I saw a banana." It will only be able to draw it. And then, even then, the person who's talking can't know. So they'll say something like, "Well, I really like bananas," or "I thought I heard a monkey sound when you were showing me a picture, so that's probably why." Okay. So that's what happens when these two hemispheres of our brain are separated. So I want to show you a quick video and I'll be quiet while it plays. It is an older video, so I apologize for the quality. This is the best quality that I was able to find, but it shows and follows a woman who is a split brain patient. So we'll watch this and we'll see from her experience what that's like.
Have a silver clip ready, please.
In the early 1960s, surgeons revived an old technique of cutting the corpus callosum to control massive seizures in some epileptics. The operation works. It also provides an opportunity to study the different properties of the two hemispheres.
Right hemisphere is gently moved away from the midline.
Vicky is a split brain patient. Because of her epilepsy, surgeons severed her corpus callosum. But now her right hemisphere can no longer communicate directly with her left.
I knew what I wanted to wear, and I would open up my closet, get ready to take it out. My other hand would like just take control. It would reach in and get something that I wouldn't want at all. You know, and then I get, I don't know, frustrated, and I throw the one on the bed that I don't want. Instead of, a lot of times I can't even hang them back up.
This antagonistic behavior between hands occurs only right after split brain surgery. It takes special testing to reveal the different processing styles of the two hemispheres. Vicky is tested frequently at Cornell Medical Center by Dr. Michael Gazzaniga.
Okay, look right at the dot.
Okay.
The tests are designed to send different kinds of information to each hemisphere.
I simply want you to describe it to me. Just name the picture you see.
Okay.
Okay?
All
So
right.
get ready and look right at the dot. Fixate the dot. Look right at the dot.
Football. 2. A woman. A person.
What else?
A girl.
Okay, I tell you what. Open your eyes. Your left hand. Right what she was doing. Close your eyes and just let your left hand go.
Vicky's right hemisphere doesn't have the language to describe the whole picture, but it can express itself through her left hand.
Good. What did you write?
Skipping rope.
Huh?
Skipping rope.
Skipping rope? No, let me show you what you wrote there.
Telephone.
Yeah, what was she doing?
Talking on the telephone.
Remember that?
Yeah.
Good. Perfect.
Vicky saw the picture on her left. It went to the left side of both eyes. The visual information then traveled to her right hemisphere. It understood the picture, but could only say, woman. Then she was instructed to write using her left hand. Her right hemisphere directed her hand to spell out the other feature of the picture. Dr. Gazzaniga asked what she had written. The left hemisphere tried to guess. It had not seen telephone, so it searched for a word somehow associated with the image it had seen. When both hemispheres saw the written word, the left, which had heard the right say woman, could now give a complete description. Woman on the phone.
In Vicky, we are able to test the separate functioning of each hemisphere by the special kind of lateralized testing that you saw. In particular, in the case where we present visual information in her right visual field, which is over here, that information is projected in her left hemisphere, which is the normally dominant language and speech system in you and me as it is in Vicky. That means if I flash a letter over here, she can normally name it, a picture, or anything else. However, when you flash the same kind of information in the left visual field, which goes to the right hemisphere, you find that Vicky is not able to easily name it because of the structure having been sectioned. The communication between the two half brains has been eliminated. How about another one here? You're doing just perfect. Oh, this is a good one.
I don't know how.
Both hemispheres respond to the words. The left triggers her smile, the right makes her clap.
So what did you see that time?
Both sides receive the command at the same time, but the left side can communicate its awareness.
So you can see how, Vicky in many ways is sort of surprised by what's happening to her body. She talks about, she knows what she wants to wear, and so she reaches into the closet, but then her other hand kind of takes over and picks something else out, as though there's another person inside of her mind that she isn't aware of, that, doesn't have the ability to talk or talk very well, and that usually goes through the other half of her brain to be able to communicate with her. So very interesting in how important the corpus callosum is to our everyday functioning. Obviously, Vicky's is doing much better because she is not having her severe epilepsy. And as the video notes, that antagonistic behavior between sides kind of goes away after the first, little while, maybe a couple weeks or something like that. And then you get used to it and you are able to adapt. And that's what also makes our brains so interesting, is that we have this capacity to adapt to changes, which is also known as plasticity. So our brain can change, reorganize, adapt, and just be able to function despite different problems or different changes that we might experience. So there's two main types of plasticity that we see. One is structural plasticity. So if we experience, an experience or we have memories, that does change our brain's physical structure. This is what people mean when they say that trauma, for example, rewires the brain. Many types of experience changes the physical structure of our brain, and that changes the way that we behave over time. However, in Vicky's case, we're talking about more functional plasticity, and that's where brain functions move from a damaged area to an undamaged area. Okay, so neuroplasticity is the most robust when we're young. When we're very little, we tend to have a whole lot more capacity for our brain to adapt to different changes. And that's because our brain is changing all the time. Okay, when we're kids, they're constantly changing and adapting. Okay, our nervous system is the most pliable or plastic when we're young, but even with that being in mind, it's still capable of incredible changes later in life as well. Okay. So now let's think about the different parts of the brain. The way we think that the brain evolved over time is really from the inside out. So, the most interior part of our brain, according to this triune brain theory, is the most primitive part of our brain. Okay, and we call that our reptilian brain or our lizard brain, if you've ever heard that before. That's more instinctive. It's more, just literally being alive and being able to breathe and have our heartbeat and stuff. Then you have the next layer, the limbic brain, that middle brain part, okay? And that's more emotional or feeling. So it starts to attach meaning to some of the experiences that we have, whereas the reptilian brain doesn't do that. And then finally, the last layer is the neocortex, which we can call the wizard brain, right? It is the rational or thinking brain. It's the part of our brain that comes up with theorems and symphonies and stuff like that. Okay, so these three layers all kind of work together, but they all have different functions. And because of that, I want us to go through each part in sort of sections. There's a little bit broader sections than what's listed in the triune brain theory, but it gives you kind of a sense of what we're going to be looking at as we go through these different anatomical regions of the brain. So we start with, again, that understanding that we have that triune brain, right? The forebrain, the midbrain, and the hindbrain. Now, these are not perfect replicas of that triune brain image, but it kind of gives you an idea of what each of these parts do. Okay, the forebrain is going to be the largest part of the brain. It contains the cerebral cortex and a number of other structures that lie beneath the cortex, which are called subcortical structures. Okay, the thalamus, the hypothalamus, the pituitary gland, the limbic system, all of those collection of structures are going to be there. Then we have this cerebral cortex, right? This most important part of our brain. Um, I mean, I don't want to say it's most important, but I think it's the most important to the way we think of our brain, the way we think of ourselves and how our brain works, because it's associated with those higher level functionings like consciousness, thought, emotion, reasoning, language, memory. All of those things are housed in that forebrain section. Okay, then the midbrain is going to have more of that limbic more of the limbic characteristics, and the hindbrain is going to be more of that like reptile style brain. So we're going to go through each of those in detail, but we're going to start with the forebrain. And the forebrain is divided into four lobes. So we have the frontal lobe, the parietal lobe, the occipital lobe at the back, and the temporal lobes at the side. Okay, so frontal lobe, parietal lobe, occipital lobe, and temporal lobes. So let's go through each one of these in detail. So step by step, we start with the frontal lobe. The frontal lobe contains the motor cortex. It's involved in planning and coordinating movement. It also has the prefrontal cortex. Many of you have heard about the prefrontal cortex, responsible for higher level cognitive functioning, judgment, and decision making. And also the Broca's area, which is essential for language production. Okay, the cerebral cortex in general and the frontal lobe in particular are associated with those sophisticated functions like planning, being self-aware. They're really often thought about as being like less primal, more sophisticated, higher parts of our brain. Okay, so other animals like rats and kangaroos do have frontal regions of their brain, but they don't have the same development in the cerebral cortices as we do. Okay, so think the closer an animal is to a human on the evolutionary tree, like a chimpanzee and a gorilla, for example, the more developed their frontal lobe is. So this is kind of the part of our brain that we think of as housing more of our humanity, the things that make us human. Okay. Within that frontal lobe, we have the primary motor cortex. Okay. One particularly fascinating area in our brain. It is this strip, you can think about it as like a headband strip going across the top of the brain. Um, it's in charge of voluntary movements like waving goodbye, wiggling your eyebrows, giving someone a kiss. That stuff is all there in that primary motor cortex. Okay. The brain loves to specialize regions. And so regions of the brain are all highly specialized. And this part is actually so specialized that you can see in terms of specific areas on that primary motor cortex associated with different body areas. Okay. And if you look at this image here to the side, this right here is known as a motor homunculus. Homunculus, it's like a little guy, okay, that shows where on the brain how much of that region is devoted to specific body areas. And you can actually see like each individual finger on your body has about as much dedicated brain space as your entire leg. Okay, because the amount of delicate, specific movement you need to be able to do with your fingers is much, much more specific and needs more brain power than your whole leg does need. Okay, your lips require about as much dedicated brain processing as all of your fingers and your hand combined. Because just think about all of the minute movements that you need to be able to do with your lips. Okay. They require a lot more processing space than just, your elbow. Okay. So this is how we can see how specialized our brain is, and we can use that motor homunculus to kind of get a sense of how much that little primary motor cortex is divided up among different parts of our body. Okay. Now, remember in week 1, we talked a little bit about Phineas Gage and the prefrontal cortex, and this is kind of the case that tells us what our prefrontal cortex is responsible for. Okay, and we talked about this a little bit. I'm going to tell you the story in a little bit more detail. So on September 13th, 1848, Phineas Gage, who was 25 at the time, was working as a railroad foreman in Vermont. Okay. He and his crew were using an iron rod to tamp explosives down into a blasting hole so that they could remove rock along a railway's path. So they dug a hole, a long, narrow hole, and they were using these posts to push the explosives down into the hole as deep as they could. Unfortunately, as that was happening, Phineas Gage must have hit something in the dirt and that caused a spark. And that spark caused the rod to explode out of the blasting hole straight into Gage's face. It went through his skull. You can see the image here. Okay. He was lying in a pool of blood with brain matter emerging from his head. About a teacup worth, they wrote down, of brain matter emerging from his head. He got up, was fully conscious, was able to walk and speak immediately after it happened, which is crazy, right? Like, that's insane. That would freak you out, wouldn't it? You would think like that person's definitely dead. They have brains coming out of their head, but he's like, "What happened? I can't see out of my eye." He's just talking immediately. Okay. In the months following that accident, people noticed that his personality had deeply changed. Okay. His friends said he was no longer being himself. Okay. So before the accident, we have records saying that he was a well-mannered, soft-spoken man, just a really nice guy. And then after the accident, he started behaving in really odd and inappropriate ways. His personality was, really inconsistent. He would be really impulsive and violent and quick to be hostile and aggressive. Okay. And this is consistent with what we now know is a loss of impulse control, which is a frontal lobe function. So impulse control, judgment and decision making, all of that stuff is in your prefrontal cortex, which is what was damaged in Gage's accident. Okay. So beyond the damage to the frontal lobe itself, we actually looked, did a little bit more digging. We have access to Gage's brain, or not his brain, but rather his skull, and we could look at the path to identify the probable damage to the pathways between the frontal lobe and other brain structures, including the limbic system. And so when the connections between the planning functions of the frontal lobe and the emotional processes of the limbic system were severed, okay, we see that Gage just became very aggressive and had difficulty controlling his emotional impulses, which is another thing that tells us about how our brain works. Our midbrain, that limbic system, is very emotional. It's very aggressive. It needs the frontal lobes to keep it in check. So when you get angry, we want to route that through the frontal lobe. So we go, yes, I'm pissed, but that doesn't mean I can just punch a guy out. I need to calm down. But when the connections are severed, it's just punch a guy. Don't even think twice about it. And that's what ended up happening with Phineas Gage. And that's one of the case studies, like we talked about in week 1, that taught us so much about prefrontal cortex and the way that the frontal lobe localization of those functions is relevant to our day-to-day life. Okay. So the other area that I talked about is the Broca's area, and you can see in the image, it's up there in the frontal cortex, kind of closing in on a little bit of that temporal lobe, but it's still in the frontal lobe. And this part of our brain is responsible for language production. Okay. It makes it difficult for us to speak if the Broca's area is damaged. Often this area can be damaged by a stroke, but it can also be damaged by, physical damage, for example. We can see there's nothing wrong with her mouth. There's nothing wrong with her vocal cords. You can follow directions. You can read, but you can't write. It just becomes very difficult for a person to communicate verbally if there is a problem with the Broca's area. So what I want to show you is another quick video. This is an individual who, after suffering a stroke, developed a lesion to the Broca's area of the brain. And so you'll be able to see how having that problem affects his communication. So I'll be quiet and we'll watch that.
Can you tell us your name?
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago.
Okay.
And
what did you used to do?
Worked Autodesk, seven sales, and worldwide, and very good. Yeah.
Okay. And who are you looking at over there when you turn your head?
That's my wife.
Okay. And why is she helping you to talk?
She's a speech.
So you have trouble with your speech?
Yeah.
And what's that called?
aphasia.
All right. And so why don't you work now?
I well, I do.
And what do you do now?
Voices of Hope aphasia.
And what is Voices of Hope?
Peterburg, and Dr. Hinckley and myself, founder for me, and I members the members probably 60 people.
So 60 people are part of Voices of Hope,
Yes.
which is an aphasia support group
Yes.
that you founded.
Yes.
And Dr. Jackie Hinckley is part of that.
Yes.
Okay, great.
It's not a support group.
No.
It's a
programs. It's 3 days. Monday, Wednesday, Friday, and and they laugh and talked. Music here, just beautiful. It's,
Great.
yeah.
Can you tell me, what does it feel like to have aphasia?
It's hard. It's it's speech. It's like words that don't understand. Brain is good, but it's speech like I don't know. It's like words, yuck.
Okay.
Yeah.
All right. Thank you so much.
Good.
Bye-bye.
Bye-bye.
Words, yuck. Okay. Speech, yuck. Hard to talk. Okay. So you can see the way that he struggles with communicating. He clearly understands the questions and he has an awareness that he's not able to produce the words very well. Okay. And this is a case of a person who has pretty decent control and capacity to communicate. He says that he actually works with a doctor to do 3-day programs for people who have this condition to help them be able to communicate better. So he's clearly doing quite well for someone with his condition, but you can see the language is stilted and he struggles to find the words to be able to communicate. Okay, so that's Broca's aphasia. All right, so now let's move away from the frontal lobe and into the parietal lobe. Okay, so the parietal lobe is also going to have a primary cortex, and there it has the somatosensory cortex, which is processing sensory information. So while the frontal cortex has that motor cortex, this has the somatosensory cortex. So we're talking about sensation. Okay, and the parietal lobe is organized topographically, just like the way that that frontal lobe is organized with that motor cortex. It also has a sort of sensory homunculus that describes the way that each part of that cortex is allocated to our body. So let's look a little bit closer at what that looks like. You can see that it comes also across the top of the head, kind of like that headband zone. And you can see that areas that require more processing power, like our fingertips, okay, the fingers are going to need more sensory awareness than, for example, your whole trunk does. You can see it takes up more space in that somatosensory cortex. Okay, and if this looks familiar to you, it is because both of those cortices are located right next to each other. They're right on top of one another. And I apologize for the quality of this image, but you can see again the way that the different homunculi allocate that information space to different parts of the body. So in the same way that the primary motor cortex has, your hand taking up more space than the entire leg, so too does the somatosensory cortex have just the tip of your finger or your thumb taking up more sensory space than the whole like trunk. Okay, so if you think about if you need to feel something delicately, you're probably not going to do it with your elbow. You're probably going to do it with your fingertips. You might bring it to your mouth to feel a texture. I know, like if I have a sliver, or I think I have a sliver in my hand, sometimes I'll bring it up to my face because I can feel it better than I can even with the tip of my finger, right? Because you have so much more space dedicated to some of those most sensitive regions of our body. Okay. So you can think about the frontal lobe and the parietal lobe as being kind of siblings. They have similar functions and they have these primary cortices that work together to give us our sensation and motor functions in our body. Okay. So moving into the temporal lobe, right? These ones are the ones that are kind of near to our ears. And happily, they actually contain the auditory cortex. Wasn't that great? Don't you love it when the brain makes sense and the thing that's close to your ear is responsible for your ear? There's another area in that side of the brain, very similar to the Broca's area. This is called the Wernicke's area, and it's really important for speech comprehension. So remember, Broca's area was responsible for speech production, the motor ability to produce speech. Wernicke's area is responsible for speech comprehension, understanding. And that makes sense because our temporal lobes are also associated with aspects of hearing and memory and emotion. So an understanding aspect of our brain is visited in the temporal lobe. So here again, you can see the comparison between the Broca's area, where we have difficulty producing language, and the Wernicke's area. This has individuals able to produce sensible language, but they are not able to understand it. Okay, so an individual with Wernicke's aphasia is essentially saying words that make sense, and they're able to do it in a cadence that is normal, but the sentences don't really make sense, and they don't seem to be able to understand what they are saying or what the other person is saying to them. Okay, so the understanding piece is gone. So the fluency is there, but the understanding is not. So the words come out as, words that are words, but they don't make sense in context. So let's watch a quick video of an individual who is experiencing Wernicke's aphasia due to an injury to that area of the brain.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment, they don't show a darn thing.
With the iPad that we were doing. Like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them.
Good.
This girl was very good and happy. And I play golf and hit other trees. We play out with the hands. We save a lot of hands on hold for us, other hands. I don't know what you get, but I talk with a lot of hand for him. Sometime. Am I talk of any more to saying?
All right. Thank you very much.
Thank you very much. I appreciate it, and I hope the world lasts for you.
Thank you. It's been a pleasure. Bye-bye.
Have a good day.
So you can see how he is you almost kind of feel crazy when you're listening to Brian because you think like, am I is he saying something? Because he has like the cadence, the normal fluency of regular language communication, but he's saying words that are real words, but they do not make sense together. Okay, so that's damage to Wernicke's area. When you're talking, just think about to yourself, what occurs for you when you're speaking. For me, I don't know if this is the case for you, but for me, I don't really put a lot of effort into talking. I just have thoughts in my mind and then they come out in my mouth, right? Like I just start talking and then talking happens. I don't really have to think about specific mouth placement, or I don't even really have to think about specific words that I'm going to say before I say them. I can just kind of get started on a thought, and then my brain is fast enough that it can have me thinking and speaking in real time, if that makes sense. And so for someone like Brian, who's experiencing Wernicke's aphasia, he opens his mouth and speaking words comes out, but he can't detect by listening that the thing he's saying is wrong. Okay. He is speaking gibberish. He is saying words that don't make sense, but it follows the right kind of cadence. It has the right sort of sound to it if you're just listening to the melody of the way he's talking. And he doesn't understand the words that he's saying, and he doesn't understand the words that anyone else is saying. And so it sounds like he's doing it right as far as he's concerned. And so, because of this area, this Wernicke's area having damage to it, he can't really understand that he's not saying the correct word. We have a little bit of an audio feedback from when we're talking. Just like if you say a word wrong, you meant to say something else and it comes out wrong, to correct yourself and say, oh, I mean rather this other thing. He doesn't have that because he can't understand spoken language. So it creates this very odd kind of experience. He seems like a really nice guy, but he'd be really fun to hang out with, but just very difficult to understand what he's talking about or what he means. And he definitely doesn't know what she's asking him. Okay. So that is the temporal lobe, major area of the temporal lobe. Now let's move into the occipital lobe. Okay, so the occipital lobe, the primary cortex in that occipital lobe at the back of our brain is the visual cortex. And so that's going to interpret our visual information. And just like the sensory motor, or the somatosensory and the motor cortex, which we call the sensory motor cortex together, is organized topographically, the occipital lobe's primary visual cortex is organized retinotopically. Okay, and that means there's a relationship between the position of an object in our visual field. So where is it? Is it in the middle? Is it to the left? Is it to the right? The position of that object in my visual field and the position of it in my cortex. So where it is represented in my brain. And this will make sense when I show you this image. So you see here, this primary visual cortex represented here on this. You can see each of these areas here within this fissure are numbered. And if we looked at it as compared to the visual field, so if you imagine your visual field as a big circle in front of you, stuff that is in the fovea, right, the center of our visual field, is towards the outside of the primary visual cortex, okay, on the right and left sides of our brain. And remember, because of that cross-lateralization, stuff that is in the left hand fovea is going to be interpreted by the right hand visual cortex. And then as you get out more, farther to the periphery of the visual field, you see that the regions are deeper inside of that fissure and may even be a little bit smaller, which again suggests how much effort and how much processing power is devoted to those regions. So you can see this massive section right here on the exterior is devoted to these two little tiny parts, whereas this relatively like smaller section right here, smaller than at least than these two, is devoted to this entire outside half of the ring. So much less of our processing power is devoted to the exterior periphery of our vision. Okay, and again, this is just an image to show you the way that our visual cortex takes in that information that is, again, cross-lateralized. So from the way that we look out in our eye. Okay. So we'll go into more detail on this specific part, how the eye works, how the ear works, all of that when we get into our sensation and perception lecture. But for right now, you can just see how those things are kind of connected and how they are cross-lateralized. So the other areas of the forebrain, which include the lobes, just the lobes that we just talked about, are the parts that are just beneath that cerebral cortex, like the thalamus and to some extent the limbic system. So we'll talk about those a little bit as well. So the thalamus is the relay center of the brain. So most of our senses are going to be routed for processing. So all of our senses, except for smell, are routed through the thalamus before being directed to all of their important areas of the brain for processing. So it's kind of the central relay center of our brain. Everything has to pass through the thalamus. Then just under that, you have the amygdala, hippocampus, and hypothalamus. Okay. And these are kind of, they're technically parts of the forebrain, but they can kind of also be grouped with the midbrain. We're grouping them here for this purpose. But the amygdala is responsible for our experience of emotion, tying emotional meaning to our memories. Okay. It's essentially all about the experience, right? So when we're going through life and we have these emotions happen, usually we have to route them through that frontal cortex, just like we talked about with Phineas Gage. But the amygdala itself really doesn't care about the rational experience or what your behavior is. It's all about that emotional meaning. Okay. The hippocampus is nearby. It's and basically the essential structure for learning and memory. It's going to hold on to our memories. And because the hippocampus and the amygdala are so close together, it creates that emotional meaning with our memories. So those two things can happen at the same time. We can link emotional things to our memories. So that's why we can have memories that bring tears to our eyes or have memories that make our heart pound, things like that, even though nothing's really happening in real life, it's just in our memories. And then finally, the hypothalamus is really kind of important for maintaining homeostasis. So regulation of blood, blood pressure, body temperature, appetite. It's essentially an interface between our nervous system and our endocrine system. Okay. It also is associated with the regulation of sexual motivation and behavior, but predominantly it's just thinking about it as being the baseline, keeping us homeostatic. That's its main purpose. So you can think about the limbic system as being the emotion and memory center. Okay, so our sense of smell actually projects directly into the limbic system. Like I told you before, it doesn't go through the thalamus. It's the only sense that doesn't. It projects directly into the limbic system. So that's why smell can provoke those emotional and memories that other senses can't. So like I can show you a picture of a box of crayons and you can smell it. Like you know that smell immediately. But also, have you ever been in a place and you're like, oh my gosh, this place smells just like Pizza Hut from when I was like 9 years old. Like, I know this smell, or this smells just like my grandma's house. Like, and you just can't quite put your finger on it, but it's just a very strong smell memory. That's the reason why, is because of the way that our smell is projected directly into this emotional memory system. Okay, so now let's move fully into the midbrain. Okay, the midbrain is comprised of sort of structures located deep within the brain, and it's kind of between the forebrain and the hindbrain. So you can actually see here how the thalamus is right here. So, like I was saying, technically that limbic system is categorized as being in the forebrain, but it's so close to the midbrain that we can kind of think of it as being a part of the lizard brain more than a part of the wizard brain, if that helps. Okay, so the two main areas of the midbrain that I want you to be aware of are the substantia nigra, which is Latin for black substance, and right next to the VTA, which is the ventral tegmental area. Those contain cell bodies that produce all kinds of neurotransmitters, specifically dopamine. They're really critical to movement. So we really need to be able to have this functional so that we can operate our voluntary movement systems. And we think that when the substantia nigra and the VTA areas are degenerated or have injury to them, that's what can kind of cause Parkinson's disease. So Parkinson's disease associated with some involuntary movement. We also see that they're somewhat involved in mood, reward, and addiction. So there may be some issues with the substantia nigra and the VTA as it relates to substance abuse disorder. And we're still kind of learning and figuring that out. The other area I want you to be aware of is the reticular formation that's centered in the midbrain, and it basically regulates our sleep-wake cycle, arousal, alertness, and some motor activities. So it actually extends up into the forebrain and down into the hindbrain, the reticular formation. So kind of is connecting both of these regions together. So it's important for a lot of different functions. If you have damage to your reticular formation, you're going to have a lot of difficulty in, being awake, being around, having arousal, meaning being alert, and being able to have voluntary motor activity. Okay, so that's the midbrain. Now let's move into the hindbrain structures. Now, these are essentially like extensions from the spinal cord. This is what we're talking about. We're talking about the most primitive part of our brain because these areas of our brain are essentially just responsible for keeping us alive. Okay, so the medulla, pons, and the cerebellum are housed in the hindbrain region. The medulla is going to control breathing, blood pressure, heart rate. It's basically just the automatic processes of the autonomic nervous system. So you don't have to think about breathing or your heart rate or anything like that. The medulla is going to control that. Okay. The word pons actually means bridge, and it serves to connect the hindbrain to the rest of the brain. Okay, it's involved in regulating brain activity during sleep. So we see that people who have damage to their pons will tend to have violent thrashing while they're sleeping because they can't really turn off their motor experiences during sleep. And so the rest of the brain is just doing rest of the brain stuff, even if they're unconscious. It's not able to regulate those functions. And then finally, the cerebellum is at the back of the brain. The cerebellum means little brain, and it literally looks like an extra little brain on the back of your brain. That one receives messages from your muscles, tendons, joints, structures in our ear, which we're going to talk about in the sensation and perception unit, that control balance and coordination and movement and motor skills, all that stuff. It's also something that we think is important for processing certain types of memories. So not all types of memories, most of those are going to be in the hippocampus, but some types of memories are housed in the cerebellum. In particularly, we think procedural memory. So those are things like how to walk, how to talk, how, specific, like, when they say that once you learn how to ride a bike, like you always know how, no matter how long it's been, or people who know how to play the piano, they can kind of do it by muscle memory. That is in the cerebellum, we think, rather than fully in the hippocampus. Okay, so we've just gone through all of the different kind of anatomy of the brain, the important anatomy of the brain. And now let's talk a little bit about how we can visualize those areas. Okay, so there's a lot of different brain imaging techniques that we use. I'm going to talk about a few of the major ones. So we're going to talk about computerized tomography, that's CT scanning, electroencephalography, EEG, functional magnetic resonance imaging, which is fMRI, and then just magnetic resonance imaging, MRI, and then positron emission tomography, which is a PET scan. And I'll show you some pictures of what those look like. So we start with the CT scan, computerized tomography scan. This uses radiation, okay, and it involves taking a number of X-rays of a particular section of your body, or in this case, your brain. The X-rays are going to pass through the tissues at different densities and different rates, which is what allows a computer to reconstruct the overall image of the part of the body being scanned, in this case, the brain. Typically, we use CT scans to determine whether somebody has a tumor or maybe significant brain atrophy. As you can see on the slide here, a tumor was localized in the image on the right-hand side. So the left side shows sort of a healthy brain, and the right one you can see in comparison has that tumor that they have isolated there. Next type is PET scanning, positron emission tomography. This one also uses radiation, and it creates pictures of the living active brain, which is exciting. So what happens is an individual who's going to receive the PET scan will drink or maybe be injected with a mildly radioactive substance, which is called a tracer. And once that's in the bloodstream, the amount of tracer in any given region of the brain can actually be monitored using the scan. And as that brain area becomes more active, more blood flows to that area. So just like when you're working out a muscle, if you're working out a specific muscle, more blood is going to flow to that area. The same thing happens in your brain. And the computer can monitor the movement of that radioactive tracer, and create sort of a map of the active and inactive areas of the brain based on a given behavior, or just sort of laying there, whatever the case may be. So PET scans don't show us a lot of detail, so they're not able to pinpoint events precisely in time, and they also require the brain to be exposed to radiation, which is bad. So we tend not to use PET scans for brain imaging. We tend to use fMRI instead. But if it's combined with CT, PET scans are still really effective and helpful in certain circumstances. So CT and PET scans still allow imaging of activity in neurotransmitter receptors when we're looking at people who are schizophrenic. There are certain areas that we're able to see. By using that hybrid. Actually, the most common way that a PET scan is going to be used is in cancer diagnostics. So, if you've ever had cancer before, somebody who has had it before, they can use this to identify areas of high metabolic activity, which tends to be where tumors are. So it still has a function from a medical perspective, but it's a little bit less effective in research for brain imaging. So MRI and fMRI, a little bit different. They actually don't use radiation, they use magnetic fields. So in just regular MRI, a person is placed inside a machine that's going to generate a really strong magnetic field. And that field causes hydrogen atoms in their body to move. Okay. So when the magnetic field is turned off, the hydrogen atoms emit electromagnetic signals as they return to their original positions. So tissues of different densities give off different signals, and then the computer can interpret and display that on a monitor. So it can kind of figure out the different densities based on the different signals that are given off when those hydrogen molecules, or hydrogen atoms rather, return to their original position after having been moved by the magnets in the machine. Very similar thing is happening with functional magnetic resonance imaging, but it actually can show changes in brain activity over time by tracking blood flow and oxygen levels. So in the same way the PET scan was exciting because we could see where the blood was going, we actually can do this better with more detailed images of the brain structure and better accuracy in time. So we can look, and you can see here in the image on the slide, when the person is being shown a figure that moves, different parts of their brain light up compared to when they're being shown different lines and patterns. Okay. So they have a much higher level of detail. And so it makes it easier for us to compare the brains of like healthy individuals with people with psychological disorders. And that gives us more of a sense of what structural differences might exist between these populations. So we use fMRI quite a bit. The final one is EEG, electroencephalography. We use this quite a bit. It doesn't give us an actual picture of the brain, as you can see, but it does give us a sense of the brain's activity. So without, we don't necessarily need information about localization, right? Where in the brain is it happening? But we may just need more timely information of when it is happening. And so we might use an EEG then. So what we use is an array of electrodes. You can see here in the image, they're being placed on the person's scalp. And they receive signals from electrical activity or brain waves in the brain. And that shows the frequency, which is the number of waves per second, and the amplitude, the height of the wave in these recorded brain waves. it's very accurate. Within milliseconds, we get extremely accurate detail. So this is important for people when we're studying, for example, sleep patterns. Okay, so there are people who experience absence seizures, and sometimes they'll experience them while they're asleep. So an absence seizure does not involve the presence of physical movement like we think of with epilepsy when, on a day-to-day basis when we think about it. This is involving a sort of disconnect, from reality where a person kind of spaces out and can't get out of it and doesn't really know what's happening. Now, if that happens when you're asleep, it becomes really difficult to report to your doctor that you're having this type of experience. So when we put an EEG on, we can see when the normal brain waves, and you can see here in this image, when the normal brain waves break away into waves that we can identify as being absence seizure related. So we can give a better diagnosis to this person based on that. Okay, so from there, you should be able to now kind of know the different imaging methods for the brain and also be able to kind of explain what the image might look like that we would get out of all of those different methods. Okay. So the final section is the nature and nurture section. We often in psychology just think a lot about how we become who we are. And we know that there's so much biological component to it, as we've discussed the different areas of the brain and all the different things that they do, but there's so much more as well that comes into it. We think that the way that we're raised and the experiences that we have also impact our nature as well. So we want to have a look and see what psychology has identified so far as being possible implications in this debate of like, do people become who they are just because of their genes, just their nature, or do people become who they are because of their environment and the way that they're raised and the way they interact with people? So let's talk about the evidence on both sides. Okay. The question is, do your genetics and biology dictate your personality and behavior, or is it your environment and how you were raised? A few of the different tools that psychology has used include adoption study, twin studies, and some things called quantitative genetics. So we're going to talk about each one of those in detail. So starting with twin and adoption studies. When we want to examine whether or not it's genes or its environment that's influencing behavior in individuals, it would be really helpful if we could compare two identical people who maybe were raised in different environments. And there are actually some people who are identical clones of one another, and those are twins. So we love looking at twins to see how they might differ based on the way that they were raised or based on their identical genetics. There's only certain types of twins that are identical, and that's where we have to kind of talk a little bit about the different types of twins that exist. So we have monozygotic or identical twins and dizygotic twins. So what happens with identical twins is that one egg is ovulated and then one sperm fertilizes that egg. And then during cell splitting, during the cell multiplication process, it divides into two. So after fertilization, black magic happens. We don't know why it happens. Some of those cells can just spontaneously divide into two genetically identical people. These two people there's no other genetic information. They are not regular siblings. They are clones. They are genetically identical and born at the same time. So because of that, they can only be same sex. They can either be two males or two females. Identical twins have the same likelihood in every single type of person. You can't be more likely to have identical twins or less likely to have identical twins. And they just are a rare occurrence that we don't know why it happens. Fraternal twins are more common. This happens when a woman hyperovulates. So when she produces more than one egg at one time, and both of those eggs are fertilized. They're going to be fertilized by different sperms. So two eggs, two sperms create two babies. These two twins are only twins by virtue of the fact that they're being born at the same time. Other than that, they're just the same as any regular siblings. So for those of you who have a brother or a sister, if you were born at the same time, you'd be twins. There's nothing extra different, extra different than having a regular brother or sister, just being born at the same time. These are called fraternal twins. And because they're made with two separate eggs, they can be the same sex or opposite sex. Okay. So fraternal twins are just regular brothers and sisters who have been born at the same time, and identical twins are clones that are born at the same time. Okay. Exactly the same genetic code. Fraternal twins are slightly more likely to occur, and you can be predisposed to making fraternal twins if hyperovulation runs in your family. So, if you have lots of twins in your family on your mother's side, so it only passes through women, and lots of women have twins in your family and you're a woman, then you can be more likely to have fraternal twins. It's also slightly more likely if you're a little bit more advanced in age, or if you're from certain racial groups, or if you are have a certain BMI. There are some things that you can make fraternal twins more likely because some of those things can increase the likelihood of hyperovulation. Okay, this is also the case when people use fertility assistance methods like IVF or medications like Clomid increase the risk of hyperovulation, which increases the risk of fraternal twins. It's not a risk, it just increases the likelihood of fraternal twins. There's nothing you can do to increase the likelihood of identical twins, though, because it's just a random thing that happens. Okay. So identical twins and fraternal twins are both very interesting to psychologists because, first of all, they're born at the same time. So that means they're going to be raised in the same way if they're kept together. So identical twins are going to be given the same types of food and the same types of clothing and the same types of attention from their parents, the same type of teacher, the same type of period of the world, the same access to, the same access to things, the same exposure to other things. All of those things are going to be the same because they're born at the same time in the same hospital in the same way. So we can keep those things as same as possible, just like we were talking about with controlling those variables last week. Identical twins are especially interesting to psychologists because not only are they born at the same time so we can control the environment, but they are identical genetically. So we can see how much is associated with environment and how much is associated with genes if they are raised separately. So what did we find? It turns out that genes play an incredibly important role in the similarities of separated twins. There was a study called the Minnesota Study of Twins Reared Apart. Now, back in the olden days, it was pretty common for people who had twins to adopt out one or both of their twins because having two babies at the same time is a lot of work. And so what would happen is if you have identical twin children, one child is raised by one family, one child is raised by the other family, you basically have two identical people being raised in two different environments. And that creates two groups, genetic relatives and environmental relatives. So we can see how much the environmental relatives have an impact on the behavior and mind of an individual compared to his genetically identical sibling. Okay. Non-twin siblings who grow up together do not resemble one another in personality necessarily. I think any one of us who have siblings, like, just think to yourself, raise your hand in your mind. Like, if you have a sibling, are you guys like exactly the same frickin' frack? Like, for most of us, that's not true. I know for myself, that is not true. I have three sisters and we are not, we are not very similar at all. However, identical twins, even if they're raised apart, have shocking similarities in their personality. So, one of the twins sets that were studied in the Minnesota Study of Twins Reared Apart were called the Jim twins. And they were shockingly similar. By the way, this photograph was taken. They did not tell them to dress that way. Like, they just showed up in the same color shirt, with basically only small differences in their hairstyle. Okay. Here are some of the things after extensive testing that they found were similar with the Jim twins, who again were raised completely separately their whole lives and never even knew they had a twin until they were adults. So both of them had beloved childhood dogs that they named Toy. Both of them preferred math and woodworking. Both struggled in spelling. Both married twice. Both of their first wives were named Linda. Their second wives were named Betty. They both had a son and both named them James Allen. They both were heavy smokers of the same brand of cigarettes. They both drove a light blue Chevy. They both had similar jobs. One was a police officer and the other one was a security officer. They both vacationed every year on the same 3 block strip of beach. They only lived 40 miles apart and they had almost identical medical history, brain scans, and personality tests. These two people who had never met each other before. And they're not the only ones. There were dozens and dozens and dozens of twins in this Minnesota study, and they had very similar outcomes where they were all personality-wise extremely similar. And when they polled those twins, they asked them, how close do you feel to your twin? Now, compared with dizygotic twins that were separated at birth, we see that monozygotic twins, 80% of them stated that they felt that they were closer than best friends and more familiar than best friends upon first meeting. And this was not the case for twins with their adopted siblings or even non-identical twins. So additionally, when we look at the way that, not only the way that they feel about one another, that they feel closer and more familiar than even their best friend when they first met, we can look at their IQ correlation. So taking IQ tests, we can see how closely related identical twins who are raised apart are in their IQ correlation. Now, just to orient you to this graph, this shows the types of relationships they have on this side and the amount of genes shared on this side, and then the percent correlation at the bottom. This doesn't mean more or less intelligence, just how similar each of them were. Okay, so in adoptive parent-child pairs or similarly aged unrelated children who are raised together or adoptive siblings with no genes shared, we have very little correlation between IQ. Okay, with half siblings who share only 25% of their genes, it's less than half correlation. However, we start to creep up as we start to have more and more genes shared, culminating with identical twins who were raised apart have a greater than 70% correlation in their IQ. And that suggests that intelligence quotient is related to our genes more than it is related to our environment, because these people who are identical clones who are raised apart have still very similar IQs. So just to let you know, there are some areas in which the study was criticized. Okay, so in more recent years of the Minnesota twin studies, the twins might have known about each other just because, if you know you're a twin or you might have known the other person existed. There's DNA testing and stuff that you can get in contact with people. So that they might influence each other to be more similar. Coincidences do happen. Some randomly chosen pairs of people will have similar traits. And typically, if we were in the classroom, I would just pair you up randomly and ask you to figure out all the things that you have in common. And probably you will have many things in common, even totally randomly. Also, the environments might be similar. So it could be the case that the adoptive families are more similar than randomly selected families. Okay, so the people who chose to take in the twin may be locals and have similar characteristics. And so it could be the case that they just happen to be raised in similar environments. But none of these factors can fully explain better than a genetic explanation why fraternal twins have more differences than identical twins. So even if all of those three things were true, we would have expected that people who are fraternal twins would have the same issues. So they might have known each other, or they just might have coincidences, or they might have had a similar environment as well. And yet, fraternal twins have more differences than identical twins. Okay, so across the board, despite being raised in different homes, identical twins were substantially similar in personality, styles of thinking and relating, abilities, intelligence, test scores, aptitudes, interests, tastes, fears, brainwaves, and even heart rate were found to be substantially similar in identical twins. So there is a really large component that our DNA, our nature, the things that we're born with, really do have an effect on our personality. So let's think a little bit more about how human genetics impacts things. So remember, just to remind you as a refresher, the theory of evolution by natural selection says that organisms that are better suited for their environment will survive and reproduce. And so those genes will be passed on, whereas individuals who are not well suited for their environment will not pass on their genes. And so genetic variety will contribute to our ability to adapt to our environments. However, there can always be mutations. Mutations are almost all of the time either neutral or bad, but sometimes they can be helpful and cause us to be more well adapted to our environment. And that means that that mutation will likely survive to be passed on. Okay. So that's the basic, foundational understanding of evolution by natural selection. But what we've started to understand is that there are relationships between our genes and our environment that can cause our genes to be expressed in different ways. Okay. So all genes set sort of limits on our potential, but our environment determines how much of that potential is achieved. Okay. One thing that we can think about with gene environment interactions rather is that it's a metaphor. The idea is that genetics loads the gun, but environment pulls the trigger. So if you don't have an environmental trigger for a gene, it may not be expressed. This field of study, where we look at the way that environment impacts our genes, is called epigenetics, and it helps us to understand how people with similar genes can have different results. So here's a quick example of what that might look like. So we have Jennifer and Karen, who are identical twins. They both have genes that absorb fats twice as fast as the average person. So they both have some kind of gene mutation. Now, Jennifer chooses to eat healthy, low fat food. And so her obesity genes are not triggered. But Karen chooses to eat fattier and unhealthier foods, so her obesity genes are triggered. So even though these two people are identical, they still have choices they can make, and they still have environmental predispositions. They can cause them to either trigger a gene or not trigger a gene, and therefore they end up with different circumstances. So this slide, we won't go into a whole bunch of detail because you're not required to know all of these mechanisms, all of these epigenetic mechanisms and how they can impact, histone tails and all that stuff and DNA methylation and whatever. But the idea is that the epigenome, which is the chemical compounds and proteins that attach to our DNA, okay, things from our environment that connect to our DNA can be altered through various experiences and environments. And those things can happen before we are even conceived. So they can happen in our mother or our father that can create changes, epigenetic changes to the sperm and the ovum that causes a change at conception. They could happen during developmental exposure. So either when we are gestating in our mothers or as children or during puberty or even late onset throughout our lives that can cause transgenerational effects, meaning these changes can occur to us before we are even conceived or at any time during the developmental process, can make changes in the way that our DNA is expressed, and that can cause effects to our children and their children down the line. So again, this is not a DNA change, this is an epigenetic change from being exposed to things like smoking or air pollution or EDCs or diet, stress. All of these things are present in the environment, and the environment impacts the way that our DNA is expressed and the way that it is passed on. So when I ask you the question, is it nature or is it nurture? The answer is it's both. So our environment can create epigenetic modifications that change gene regulation, which produces a different phenotype, right? What is expressed. And our genetics have various sequence variations that affect gene regulation and therefore the phenotype. So while most research suggests that genetics is predominantly associated with every single part of our life, environment also has a major role just because it can also modify the DNA sequence. Okay. So another, we're not here together in the classroom, but if you had to pick, just think to yourself, what would you think is more influential? Do you think it's nature or is it nurture or why? And then think about what you think. If it's nature, nurture, or if it's a perfect blend of both, what implication does that have on the way we advise people as they live and grow and change over time? Because if we really think that people are just set in stone from birth in their genes, then there's no point in trying to change things, right? But if we think that people can change, that environments can make a difference, then there is a lot of potential for growth. And that's what the science really seems to demonstrate as it relates to psychology. Okay, so let's review this second part. We talked about the parts of the brain, how they can be divided into lobes, what roles each major brain region plays, and what's meant by the lizard brain and the wizard brain. So, what do we mean when we sort of divide up parts of the brain into those categories? And then we talked about nature, nurture, and epigenetics, how they influence personality and behavior. Okay, so that is the end of this unit, the biopsychology unit. Of course, if you have any questions, I would be happy to receive those in my inbox. I would love it if you got together and had a little study party and talked about the slides. Make sure to review those and that you understand from the reading and from this lecture what you're supposed to have learned about this unit. And again, I'm here to answer any questions that you might have. I'll really quickly go over the assignment for this week with you, so bear with me. So here you can see the ALA for this week, which is the 3D brain simulation. So you're going to go to this link. It has a 3D interactive model of the brain, and you can actually rotate it around and kind of get a sense of what all of these things look like. Okay, so the first thing you're going to do is you are going to use the cursors to rotate the brain around. You're going to use these labels buttons to on the actual 3D model to see if you remember the names of the cortical lobes that we talked about during the lecture. So I want you to guess what it is, tell me what you guessed, and then look and see if you got it right, and then tell me what you put. Then you're going to label each part with here. So you just click there, type here. So this is why it's really important that you're downloading the worksheet and opening it in Word so that you can fill in all of the different parts. Okay, in the next part, you're going to switch to the brainstem view. You're going to scroll down, look at the selector, turn to the pons subsection. Everything is very laid out, all of the specific directions that you need to follow. Read the descriptions and then answer these specific questions. Then I want you to take a screenshot of the labeled anatomy of the brainstem and replace this blurred placeholder image. And you're going to do that for each one. So you're going to do that for the frontal lobe. You're going to do that for the limbic system. Oh, it looks like I accidentally put frontal lobe here. This is supposed to be the limbic system. Don't get tripped up with that. And you're going to answer these individual questions so that you basically at the end of the day have this worksheet that contains all of the parts of the brain, all of the important functions about it, interesting case studies that help you understand how it works. And then finally, you're going to watch this short video about neuroanatomy and answer these reflection questions. So specifically, I would love for you to tell me your favorite part of the video and the timestamp and the quote from that part of the video so that I can find it, so that I can enjoy it as well. Okay, so all of that is going to give you this really great and helpful neuroanatomy study guide at the end of the day. So I hope that you will put a lot of effort into this because it's going to be really helpful for you to learn the parts of the brain really well so that when we start talking about sensation and perception and consciousness and intelligence, I'm going to be able to refer to a part of the brain and you're going to know exactly what I'm talking about. Okay, so if you have any questions about this or any part of the lecture, again, please don't hesitate to reach out. I hope you all have a really safe and fun holiday, and I will see you next Saturday. So take it easy, everybody, and I will see you then. Bye.